During this presentation, we will describe the main physiological and biomechanical differences between the walking and running of a participant.
Cardiovascular system. The purpose of the cardiovascular system is to distribute oxygen and energy around the body, removing waste products such as carbon dioxide. Here is a simple representation of the cardiovascular system. Heart rate multiplied by stroke volume equals cardiac output. Cardiac output is the volume of blood ejected from the left ventricle per minute. This all depends on preload, contractility, heart rate and afterload. When exercising, the heart rate increases in beats per minute. The heart. Arteries are blood vessels that always carry blood away from the heart. Veins are blood vessels that always carry blood to the heart.
Heart rate is proportional to the intensity of exercise. As the intensity of exercise increased, the heart rate increased. With regular aerobic exercise, the resting heart rate of a person will drop as the heart becomes stronger. This is called cardiac hypertrophy. A lower resting heart rate is a sign of good health. The heart rate monitoring equipment is dampened and applied to the participant at the base of the sternum. The participant is seated and relaxed while two investigators conduct the test. One investigator records the changes in heart rate of the participant every 30 seconds over a 3-minute period, while another investigator keeps a track of the time to ensure the results are taken at the correct time. We then did the same process with the participant walking for a 3-minute period at 4.5 kilometers per hour. And finally, we tested the heart rate of the participant when running over a 3-minute period at 9 kilometers per hour. These are the heart rate results we recorded. The table shows the resting heart rate was at a healthy 70 beats per minute. Then, as the intensity of the exercise increased to the 4.5 kilometers per hour walk, the participant's heart rate increased to about 86 beats per minute. The speed was then increased to 9 kilometers per hour, and after 3 minutes, the heart rate rose to 117 beats per minute.
Blood. Blood is living cells suspended in plasma. Plasma, leukocytes and platelets, and erythrocytes are the main components of blood. Blood is used for transportation, regulation, and protection. The regulatory functions are homeostasis, to maintain body temperature. The chemoreceptors maintain the pH in the body tissues, and to maintain adequate blood volumes in circulation. The protective functions are prevent blood loss through blood clotting cascade, and also to prevent infection through antibodies, complement proteins, and white blood cells suspended in the blood. Blood pressure is the force that is applied to our artery walls, measured in millimeters of mercury.
We then tested the participant's blood pressure at rest after walking at 4.5 kilometers per hour for 5 minutes and after running at 9 kilometers per hour for 5 minutes. From the results, we expect to see an increase in the blood pressure of the participant as the intensity of the exercise increases. Expose the right arm and apply cuff 3 to 5 centimeters above the point where the brachial pulse can be palpated, with the middle of the rubber bladder over the brachial artery. Rest the arm comfortably and place a stethoscope over the brachial artery below the cuff. Close the valve of the inflation pump and inflate the cuff rapidly to above the expected systolic pressure using the hand pump. This obstructs blood flow in the brachial artery. Slowly open the valve and release the cuff pressure at a slow rate. Allow the pressure to release without closing the valve again. When the first pulse is heard, memorize the mercury level in millimeters of mercury. This is the systolic pressure. Write the value down once the pressure has all been released. As the cuff pressure falls, the sounds get louder and then suddenly become muffled and then disappear. The mercury level when the sound disappears is the diastolic pressure.
As you can see from the results, blood pressure increased as the intensity of exercise increased. This is because the body is in need of oxygenated blood for the skeletal muscles, and the waste products, such as carbon dioxide and lactic acid, must be removed.
The main function of the respiratory system is to deliver oxygen to the muscle tissue and removing carbon dioxide from this tissue. This is achieved through 4 phases. The first being pulmonary ventilation. This is the movement of air in and out of the lungs. The next phase is the gaseous exchange of oxygen and carbon dioxide in the alveoli. Gaseous exchange is the transfer of oxygen from the red blood cells through the connective tissue into the alveolus and carbon dioxide into the blood. The next phase is the transportation of gases. Oxygen is carried through hemoglobin in the blood to the skeletal muscles. The final stage is the diffusion of oxygen and carbon dioxide between the bloodstream and the tissue. The conducting zone is made up of the nose, pharynx, larynx, trachea, bronchi, bronchial tree. The cleaning and warming of air entering the body occurs in these zones. The respiratory zone is where gas exchange takes place. This consists of the bronchioles, alveoli ducts and alveoli. The lungs are made up of mainly airspace, and the pleura, including a double layered wall separated by pleural fluid. This allows movement of the lungs during breathing. The alveoli are small, thin-walled inflatable sacs at the end of the bronchioles. They are encapsulated by pulmonary capillaries. They provide a thin barrier and a large surface area for gas exchange.
We tested forced expiratory volume using the vitulograph at rest after 5 minutes of walking at 4.5 km per hour, after running for 5 minutes at 9 km per hour. After analyzing the results, we discovered that there was little to no change in the forced expiratory volume at rest after walking and after running.
The participant starts by inserting the vitulograph thermal graph paper onto the machine. He ensures that the needle is placed on the 0 point. He then inserts a disposable mouthpiece onto the flow head connection tube. The participant ensures the machine is on and the needle is positioned directly on 0 and that the paper will travel to the right to ensure that the needle makes a trace on the paper. A chair is placed behind him for health and safety reasons. The participant takes a deep inspiration before maximum force expiration for 6 seconds. He is wearing a nose clip to be certain nothing is expired through the nose cavity during the test.
There are 3 types of muscles in the body: cardiac, skeletal, and smooth. Cardiac muscles make up the walls of the heart and are involuntary. Smooth muscles are located in all hollow organs other than the heart. Skeletal muscles are the muscles which are attached to the skeleton and are the muscles involved in creating movement. They are also the only type of muscle which is voluntary.
How muscles work. Muscle fibres are a mixture of red and lighter colours depending upon the fibre type composition. Muscles are striated. Factors that affect muscle action: size and shape, number of muscles, nature of a tendon at origin and insertion point, angle and place of insertion. When calcium is present, ATP can be broken down to release the energy to allow muscular contraction to occur. The motor neuron is the nerve that attaches to the muscle fibres. Each muscle has several motor units. The number of fibres that a motor neuron attaches to depends on the muscle. The fibres attached to a motor unit are either fast or slow twitch, never a mixture. A motor unit is described as a single motor neuron and all of the muscle fibres it innervates. A motor unit can contain anywhere between 10 and thousands of muscle fibres. Muscles which produce large powerful movements contain motor units with large numbers of fibres. Therefore, those small intricate movements contain only a few fibres per motor unit. Where the synaptic knobs of the neuron meet the muscle fibres is known as the neuromuscular junction. When an impulse reaches the neuromuscular junction, this starts the sliding filament theory of muscular contraction. The sliding filament theory is a method by which the muscles are thought to contract. Each muscle fibre is made up of myofibrils. These contain smaller structures called actin and myosin. Actin and myosin can also be referred to as myofilaments, as they are protein filaments found in myofibrils. Myosin and actin run in parallel to each other along the muscle fibre. Myosin has tiny globular heads producing from it at regular intervals. These are called myosin heads. These form cross bridges and perform a pivotal role in muscle action. Then a power stroke is performed. Myosin heads connect to the actin at the binding site. This only occurs when tropomyosin is out of the binding site. This is done through the release of calcium ions flooding the sarcomere. Troponin moves tropomyosin away from the myosin binding sites on actin. This effectively unblocks it. The myosin heads on the thick filaments hook onto and so pull the actin filaments towards the centre of each sarcomere. The appearance of this action goes from the transition of relaxed to fully contracted muscle. As the thin filaments slide over the thick filaments and the muscle reaches its fully contracted state. When the myosin heads on the thick filaments relax, they release their hold on the actin filaments, thereby allowing them to slide back to their relaxed positions. The all or none law basically suggests that if the impulse arriving at the motor end plate is big enough, then all the fibres attached to the motor unit will contract. If the signal is not big enough, none of them will. The all or none law applies to the contraction of fibres within a motor unit. When a motor unit activates, all of the fibres within the unit contract at full force. There is no strong or weak contraction. The strength of the resultant whole muscular contraction depends upon the number of motor units recruited. Another way of increasing the strength of a muscle contraction is by decreasing the time between impulses so that the muscle fibres do not have time to relax, resulting in a continuous wave of contractions known as wave summation. In order to increase the length of a contraction, a kind of rotation system is implemented, whereby some units contract while others rest and continuously alternate. This is known as spatial summation.
Joints. There are over 100 joints in the body, split up into 3 categories: immovable, slightly movable, and freely movable. They contain cartilage, which is connective tissue found principally at joints between bones. It reduces friction between bones and enables flexibility in joints. The main joints involved in walking and running are coxal, tibiofemoral, and taloceural. Levers. All joints in the body are categorized as a type of lever, either first class, second class, or third class. All levers are made up of a fulcrum, a point of rotation, and a source of force, usually the insertion and a load being moved.
Angular kinematics. Circular motion or rotation of the body. Speed, velocity and acceleration. Speed is a scalar quantity. It is measured on a scale of 0 to whatever, but doesn't take into account the direction the body is moving. Speed is measured as distance travelled divided by time. Motion is speed and direction, thus making it a vector quantity. Velocity is a vector quantity and is measured as displacement divided by time. Displacement is the distance travelled in a straight line.
Planes and axes. The anatomical position is a description of the body in a specific stance. It involves standing with feet together, hands away from hips, and palms facing forwards. The sagittal plane divides the body from front to back. The frontal axis passes through the body horizontally from side to side. During walking and running, the body moves in the sagittal plane and the frontal axis. The main joints involved in walking and running are glenohumeral, humeroulnar, radiocarpal, coxal, tibiofemoral, taloceural. On our participant, we chose to label the right leg.
Camera setup.
Very difficult.
To ensure the camera is set up correctly, during our video we used a 3-4-5 right angle triangle which was taped to the floor. The right angled corner of the triangle is set up at the centre of the frame. The camera is set up at hip height. For scaling we used a 1 metre ruler. The shutter speed was set at 250 frames per second for walking and 750 frames per second for running.
Walking gaits. The movement of walking is made up of repetitive cycles, each one of which is called a stride. Each stride contains 2 steps. When their right foot is in contact with the ground, it is called the stance phase. This makes up 60% of a stride. When the right leg is in the air, it is described as the swing phase. This makes up the other 40% of a stride.
The key phases of walking and running are the touchdown, the swing phase or toe off, and heel rise. At touchdown, the tibiofemoral joint becomes flexed. During the heel rise phase, the joint becomes extended. In the swing or toe off phase, the tibiofemoral joint became flexed.
This graph shows the minimum and maximum angles of extension and flexion within the knee whilst walking and running.